So thanks everybody for joining. I'd like to start by acknowledging the traditional owners of the land on which we meet, and in particular in this room, we're meeting on the lands of the Bidjigal people, and their lands stretch from down around Coogee, up the Georges River and around into what we currently know as the Hills District. I'd like to pay my respects to elders past and present, and extend that respect to any Aboriginal or Torres Strait Islanders joining us today. So today we have two presentations from people finishing their PhDs. The first of those is from Gail Cross, who's a clinician scientist. She's done a very peripatetic PhD, starting in Singapore and finishing in Melbourne. And she's done that on TB, and having supervised Gail, and without going into details, she's done it during COVID, and COVID was the least of her issues during that time. So I'll hand over to Gail now to present the work she's done over the last 3 to 4 years. Gail?
Thank you. Thanks, Tony. Thank you for the opportunity to present today. And I have the wonderful opportunity actually of being able to participate in this phase 2B trial, which was testing adjunctive Rosuvastatin for drug-susceptible pulmonary tuberculosis. As Tony mentioned, it started off in Singapore. So I was a clinician at the National University Hospital in Singapore. The trial was funded by the National Medical Research Council. And Tony very kindly took me on as a PhD student while I was there. And himself, Dr. Christina Chang and Professor Nick Payton, who works in Singapore, took me on board as their PhD student. So I'm going to present some of that data today. So an overview first of what is host-directed therapy against tuberculosis, and then specifically why would we use statins for tuberculosis treatment. I'm then going to go into some key findings from the Rosetta trial, which was the summarized name of this trial, and then a couple of sub-study data, sub-studies, the data from some of the sub-studies, so PET-CT sub-study as well as a cytokine-chemokine sub-study. So there have been many candidate host-directed therapeutic agents that have been proposed, some of which are thought to potentially be able to expand on T-cell subsets, others which have more rigorously been tested, including in humans, and have been shown to be able to control intracellular bacterial growth. Sorry, not necessarily in humans, but certainly in mouse models. And then others which are thought to potentially be able to inhibit inflammation, tissue necrosis, and fibrosis as well. So we decided, because there was substantial experimental evidence in statins, that we would like to take it forward, which I'm going to show you in here. So statins have been shown by a group led by Norton Datta, who's from Johns Hopkins, who have in a variety of mouse models have been able to show that essentially you can reduce the lung CFU of MTB lung CFU by about a log with the addition of statins in addition to standard TB therapy. But what was most interesting about the work that they proposed was that if you use it in combination with TB therapy, you might be able to actually reduce the risk of disease relapse. And why that's exciting is because one of the proposed mechanisms of being able to end TB, which is WHO goal, is really to reduce the duration of TB therapy. If we can change it from being 6 months to perhaps 2 months or even 1 month, we might be able to get closer to that goal. But we need to be able to do that in a fashion that doesn't then increase the risk of relapse. So it was quite exciting to see data that perhaps the addition of rosuvastatin or a statin drug could potentially be the answer. I don't really have time to talk about the mechanism of action, but I'd be happy to answer questions later on or speak about that in person. So in this trial, we randomized people to get standard TB therapy, which was the control arm, which was 2 months of 4 drugs, and then followed by 2 drugs for the remainder of 4 months. And in the rosuvastatin arm, participants received, in addition to standard TB therapy, 10 milligrams of rosuvastatin for a duration of 8 weeks in total. At the 8-week time point, we collected our primary efficacy outcome, which was, sorry about the acronyms, I've presented this before to a TB familiar audience, but this is time to culture conversion in sputum, which is the primary way that we measure the biomarker that we use for efficacy, especially in phase 2b trials. And then we also looked at safety of the use of statin drugs. So we collected incident grade 3 and 4 adverse events at the end of the trial, so at the completion of the 6 months as well. So the trial was performed in several countries, in Uganda, in Vietnam, as well as in the Philippines. And over the very tough period of September 2020 to January 2021, tough for all of us, we had 300 patients pre-screened, and then eventually 137 participants who were enrolled into the trial. So we collected sputa over the duration of the trial along with all the clinical evaluation markers. So this was the finding from our trial. So we found that the use of rosuvastatin at 10 milligrams over the first 8 weeks really had no effect on sputum time to culture conversion. So we used a Cox proportional regression hazards model to do this, which controlled for the randomization country. And unfortunately, both in the liquid media as well as in the solid culture, there was an increase in the hazards ratio, which favored rosuvastatin, but of course the confidence intervals crossed 1, meaning there was no treatment effect. However, in a pre-specified subgroup analysis, we really found that there might have been a difference in those who were enrolled from Vietnam as well as from the Philippines. So the hazard ratio that we achieved for the Asian ethnicity was 1.72. And once again, because we had very small numbers, this was a subgroup analysis, there was no statistical significance, but it was quite a stark difference that we'd have a hazard ratio of 1.7 for those who were enrolled from Vietnam and Philippines, but 1 from the African ethnicity. And this was particularly interesting because we know that rosuvastatin results in a twofold elevation in the median exposure of the drug in Asian people, which is why rosuvastatin can only be used at a ceiling dose of 20 milligrams, whereas in the Caucasian population, you can go up to 40 because there's naturally increased exposure. So we hypothesized, which still hasn't been tested, that perhaps the effect that we were seeing was as a consequence of increased rosuvastatin exposure. But in addition to that trial, we also did several biomarker sub-studies and collected data towards that effect. And what I'm going to show you, one of the biomarkers that we collected was a PET-CT sub-study. So within the trial of 137 participants, for reasons of cost, we did PET-CTs at pre-treatment and after the 8-week time point in 25. And these participants came from the Philippines and Vietnam. And we did essentially, I analyzed 48 scans over this 2 time point period, and it was equally balanced in both arms. I had the help of a nuclear physicist to develop a protocol for analyzing these scans. And just very quickly, in PET-CT, essentially, the typical way of measuring, the metric that's used is SUV max or SUV mean. But what we developed, which is also used in oncology, is developed a volumetric metric, which is a metabolic volume of how essentially what volume of the lung is hot and how hot is it. So that's your metabolic volume as well as your total lesion glycolysis. And what we found is, once again, adjunctive rosuvastatin has no impact on any of the PET-CT metrics measured. So there was no difference between the rosuvastatin arm and the control group for total lesion glycolysis, and for SUV max, once again, no difference between the treatment groups. However, what we did find was that TB treatment alone can cause substantial reduction in your total lesion glycolysis within a period of 8 weeks of treatment. So your TLG reduces by 64.3% in this very small study, and your total metabolic volume goes down by 56.1%. Why this is important is because most of the studies in TB up to that point in time had really used just SUV mean or SUV max to demonstrate change over time. Probably move on very quickly from this, but essentially what we then what this resulted in was the question of, can we develop PET-CT as a non-sputum-based biomarker of TB treatment efficacy in clinical trials? And we make the argument that it potentially can in a publication. So moving on quickly then to the chemokine and cytokine substudy in this trial. Although we collected blood and plasma from 137 participants, once again, for reasons of cost, we really analyzed 80 participants' data alone. So the plasma was fun to have plasma, and that was stored at -80. It was unstimulated, and IMCB in Singapore, so it was analyzed centrally from all these sites, did a 41-plex immunology assay. And then the data was analyzed by myself. And, you know, we kind of expected to find this, but you get sort of a broad immunomodulatory effect of TB treatment. So you get reductions as expected in IL-1R alpha, interferon gamma, IL-6, all the pro-inflammatory cytokines that are elevated as a consequence of a pulmonary infection like TB come down with TB treatment over time. To do this, I used a mixed linear effects model and adjusted for baseline cytokine value and time on treatment and treatment group, as well as some clinical variables, which I've shown here. So this wasn't really unexpected, but it's nice to always show something that's been demonstrated in the literature before. But what we did find that was which was quite unexpected was that there was a difference between the treatment groups, so a difference between rosuvastatin and the control group. And the difference was that rosuvastatin, which is largely thought of as an anti-inflammatory drug, in addition to being an anti-cholesterol agent, if anything, blunted the reduction in these anti-inflammatory cytokines. So rosuvastatin effectively increased, for most of these cytokines, increased the production of these anti-inflammatory cytokines, except for one, which is macrophage something chemokine. Thanks, Tony. But this was quite surprising, and we didn't understand it. But I've just graphically demonstrated here. Essentially, particularly, in fact, at the time week 4 time point, you get an increase in IL-1 beta production, an increase in IL-12, increase in TNF alpha, and then a blunting of the reduction in interferon gamma. And I haven't shown the graphs, but similarly for IL-6 and IL-17A. For example. And unfortunately, we didn't measure IL-18. And this is unfortunate because upon looking to see what could be the mechanism for this, we realized that, in fact, there is plenty of experimental evidence to show that statins, although they are largely thought of as an anti-inflammatory drug, in the setting of an infection can actually activate caspase-1. So when you get an infection with various, particularly intracellular bacteria, you get activation of the inflammasome, and you then get activation of caspase-1. This enzyme is responsible really for the proteolytic cleavage of pro-IL-1 beta and IL-18 into its active form. And subsequently, it's sort of like a gateway cytokine that opens the door to inflammation. And what has been shown in both in vitro using MTB as well as lipopolysaccharides and various animal models is that if you use a statin in the setting of infection, you really get activation of caspase-1 more than you would in the absence of the statin and increased inflammation. And I think what was particularly interesting is that there was a trial called the SAILS trial, which had 744 participants with acute respiratory distress syndrome who had a lung infection, where they showed the use of adjunctive statin increased the production of IL-18, and those with increasing IL-18 on the statin had the highest risk of mortality. And it is for that reason, in fact, that whilst there was a lot of promise for statins being used in the setting of intensive care or intensively cared for patients, it fell out of favor dramatically as a consequence of this trial. But at least I didn't know about this potential mechanism. So in conclusion, Adjunctive rosuvastatin had no effect on accelerating sputum culture conversion within 8 weeks, but there is possibly a treatment effect in Asian versus African cohorts. This is a hypothesis that needs to be tested. It had no effect on PET-CT changes, but the volumetric indices on PET-CT are better than SUV. And finally, adjunctive rosuvastatin, in fact, was shown to increase the secretion of inflammatory cytokines in the setting of TB. And we hypothesize that this may be due to enhancement of caspase-1 activity. Once again, another hypothesis that needs to be tested. I have lots of people to thank because this was, as trials are, big, complicated work with many people. But I have to, I have a lot of gratitude to Tony, as well as to Christina Chang, and Professor Nick Payton, my other PhD supervisors, and of course, the participants of this trial, without whom I wouldn't have this data.
So thanks, Gail, for that tour de force and that rapid presentation of, an immense amount of work, some of which is very hot off the press. So I'd encourage people online to access the Q&A button and ask any questions. And for people in the room, please come to the microphone so that people online can hear the questions. So if there are questions for Gail, please come forward. Gail, just thinking about the initial observation that the statins made no difference, but there's this tantalizing observation that in Southeast Asians, there's that potential effect. I know you've looked at a range of variables that modulate the cytokine effect, but did you include site of recruitment or?
We included site of recruitment. Sorry, we included site of recruitment in the Cox proportional regression analysis, so in the publication, but no other clinical variables were included, apart from not burden of disease, for example, not, HbA1c and meaning diabetes and HIV status was stratified for in the trial, but no other clinical variables, particularly not burden of disease.
So do you think it's surprising that you see that potential change in the clinical outcome, but no difference in the cytokine outcome between those groups?
I think that the difference, I think that statins, as with a lot of immunomodulatory drugs, have multiple mechanisms of action. So the way, and I might speak towards this quickly, is that the way it's thought to work to actually enhance its mycobactericidal activity is by enhancing phagoliposomal fusion. So essentially, MTB requires cholesterol, which it imports from its host in order to, it uses it as a carbon source, but it also uses it as a virulence mechanism to inhibit phagolysosomal fusion. So if you limit the amount of cholesterol, as has been shown in many, many experimental studies, you can actually reverse phagolysosomal fusion. So I think it's working to clear mycobacteria by releasing the inhibition of phagolysosomal fusion, but it's still causing an increase in the inflammatory cytokine production. And I think, as I've written in the paper that I've sent to you very recently, that I think we need to be cautious about taking statins forward, because even whilst it might clear mycobacteria, the risk is that we might actually enhance the immunopathology of TB disease as a consequence of its two different mechanisms.
Yeah, I was thinking exactly that. And, it's a shame that we don't have more data on if we'd done the PET in more people, but obviously because you might actually be able to look at that physiologically over time. So it's really intriguing. And I think it's a real lesson. So, in parallel with this study, there was a large study of statins in people with HIV, which showed, which was a phase 4 trial done globally, and that showed unequivocally that you got better clinical outcomes in people on low-dose statin in terms of cardiovascular outcomes. And, the smart doctors associated with that trial are sure that the effect is that in terms of cardiovascular outcome is in excess of what you'd expect for the cholesterol lowering dose. So they're hypothesizing that statins have an anti-inflammatory effect in that case.
Can I
So,
say something
no,
about
so-
this? So that's the Reprieve trial. And what was interesting about that, so it was built on the Jupiter trial, which was not in a people living with HIV population. So, you know, just general population, and they showed huge reductions in HSCRP as a consequence of statin group, but it was also a trial that recruited people with elevated HSCRP, highly sensitive C-reactive protein, so cytokine. And but the difference when I looked at the, you know, the supplementary data from the Reprieve trial, it was infinitesimally small reduction in high SCRP in the people living with HIV. So I can't understand how it could be as a consequence of cytokine production.
So Virginia.
Thanks for a great presentation. And I'm probably the last person that should be standing here because this is so far away from my area of research. But I wondered, for someone like me, you could maybe explain or unpack what the implications are for TB prevention or treatment, you know, guidelines, whatever it might be. Thank you.
So actually there's plenty, and I haven't put it in here, I just described the experimental data, but in fact, there's lots of epidemiological data to suggest that if you look at retrospective cohorts, unfortunately, but retrospective cohorts, huge numbers, we're talking 20, 30,000, people who receive statins are at reduced risk in an endemic setting of developing incident TB. So it's quite clear that it does have some, maybe not through an immunomodulatory mechanism, but because you're limiting the ability of mycobacterial growth, perhaps as a consequence of reducing people's cholesterol. I don't know. That there is certainly a signal, at least, of statins reducing the risk of incident TB. So prevention of TB.
So we might go online. Yeah, so so there's a question from Braulio, which is great presentation. I didn't see the clinical outcomes to measure efficacy, just microbiological. What's the rationale for that? Any chance to reanalyze the data using clinical outcomes? Yeah,
we collect a lot of clinical outcome data in this trial, and we didn't analyze it because really the clinical outcomes were, the microbiological outcomes were very good. So, you know, we had a huge proportion of the participants who culture converted anyway at the 8-week time point, which is your surrogate marker of treatment efficacy in general. And we, you know, the clinical outcome data, I'm not such a strong believer in it, unfortunately, because, you know, you collect things like fever and cough. They're quite soft sort of markers of how people improve, and which is why we didn't necessarily put it in the paper, but we certainly have that data and we could go back to it if needed. Yeah.
Indigo.
Thanks. It would the effect would be different if it was an extrapulmonary TB, like, for example, and even miliary TB, instead of having large cavitation on the apex. And the second question is, what stage of the disease were the patients? So, like, do they have an advanced post-primary TB, or they had primary TB? Or?
So in response to the first question, what do I think? I think from a mechanism perspective, there shouldn't be any difference in how a statin would work to treat extrapulmonary TB. Having said that, though, almost 90% of the studies we do is in pulmonary TB because we don't have a biomarker of how we measure treatment efficacy aside from sputum culture conversion. There's no blood test, there's no urine test that tells us if someone's getting better. Clinically, people just have stop having a fever, feel better, have more energy, put weight on, these very soft markers. Lymphadenopathy. Yeah, so I don't dispute that there's huge proportions of people with extrapulmonary TB, but in a, from a scientific sense, being able to quantify how they get better from it is just difficult. And I wish we had a blood-based biomarker. And in answer to your second question, which was, can't remember already. Oh yeah, so huge burdens of disease. So it was, upwards of 50% had cavitation in their lung. And we also did this measure of what proportion of the lung was affected. And once again, 50% had 50% or more of their lungs affected. So yeah, very, very different to Australia, where we see minimal disease typically. Yeah.
Okay, if there's no more questions, just thanks, Gail, for a great presentation. And I'll hand over to Nico and Lok.
Thanks for coming, everyone. Our next presenter is Lok. Lok is a PhD in his final year of PhD, co-supervised by me and Rowena Bull. His project mostly in immunovirology and vaccinology. And he's a clinical microbiologist and infectious disease physician from Nepal. It was absolute pleasure having him. He was absolutely fantastic supervising Lok, and I hope we'll keep him longer. But he will finish his PhD.
Thank you, Nico. Thanks for supervising me and taking me in this university. Thanks everyone for joining me to listen about this one. My topic of my presentation, today's presentation, it is the bivalent Omicron vaccine. And I know you guys and we all are fatigued with the COVID, and maybe we don't want to hear about it, but it's my PhD project, so I don't have a choice. Okay, I'll start with a bit of virology of SARS-CoV-2 viruses. These are spherical crown-shaped viruses, which are 60 to 140 nanometer in diameter. And the genome is a positive sense single-stranded RNA genome, and it is 29,903 base pair long. And the genome, they encode for 4 structural proteins like envelope, spike protein, membrane protein, and nucleocapsid proteins, and several non-structural proteins. And the spike protein is the main structure of this virus. It gives it's the typical appearance, and it is also necessary for their entry into our cells. And we also quite love the spike protein because we made our vaccines against this spike protein and also all the antibodies. And a bit more about the spike protein. There are 2 parts, the S1 and the S2 subunit. They are joined by the purine cleavage site. And the S1 subunit, they have the main structures like the receptor binding domain and receptor binding motif. And they bind to the ACE2 receptor in human beings and enter into our cells and cause their effect. And this figure, it shows 2 stages or 2 states of the RBD, the open state and the closed state. And the objective of this particular project, the objective was to compare the neutralizing antibody, memory B cell response, T cell response between the monovalent, the typical monovalent vaccine, the Pfizer or Moderna, and the bivalent vaccine containing spike of ancestral, the Wuhan, and the Omicron BA1 vaccine. And it is from Moderna. The number is, this is the number. And the study design, it is a very, a big collaborative study, and it was funded by NSW Health. And it is a collaboration between University of New South Wales, University of Sydney, Westmead Hospital, and the Garvan Institute. And this is the study design. We took this 31 subjects from the big BEAM cohort. And then among these 31, 18 of them received the bivalent vaccine, and the 13 received the monovalent vaccine. And we took the sample at 2 time points, like before the booster and the around 1 month after the booster. So I'll be calling it pre-booster and post-booster in my result section. Methodology, we had a big team and we divided our tasks on the basis of that. The virus neutralization test, it was performed by Stuart Turville and his group. B cell assay, I did the B cell part with the supervision from Professor Rowena Bull, Professor Nicodemus Stedla, who is here, and Professor Andrew Lloyd. And the T cell assay, it was taken up by Professor Tony Cunningham and his group from the Westmead Hospital. Now I'll start with the results from the virus neutralization test. For the virus neutralization, Stu and his team performed rapid high content SARS-CoV-2 micro-neutralization assay with had 24 cells. 24 cells, 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 And this were the results. Firstly, the neutralization titer was increased post-boost in both the monovalent and the bivalent group. This magenta is the bivalent and the gray is the monovalent. I will use the similar color for all my graphs. The neutralization titer was increased post-boost in both type. But when we go to the individual, like pre-boost and post-boost, when you look at them carefully separately, what happens is that the pre-boost, there was no statistical difference between the ID50 for each variant between the monovalent and bivalent vaccine. Whereas in the post-booster group, there was significant difference in the neutralizing antibody titer for all the variants, including the JN.1. And the JN.1 was not even present at the time of the sample collection. So it was quite interesting to see that. And I forgot to mention something. Oh, okay, it's not here. Okay, and the effect of some of our subjects were infected before the booster during the time, but some of them were not. And we wanted to see if the infection affected our neutralizing antibody titer. But when we did the statistical analysis, the prior infection status did not change the neutralizing antibody titer between the monovalent and bivalent vaccine. And we have also done the multivariate analysis that I will be showing at last. And the B-cell assay, for the B-cell assay, we did spectral flow cytometry with the receptor binding domain of 4 variants as antigens, that is Wuhan, Delta, BA1, and BA5 variants. And we wanted to detect memory B-cell response against the individual RBDs and also against the multiple RBDs. And as we can see here, both monovalent and bivalent vaccine groups, they significantly boosted RBD-specific memory B cells against all 4 variants. But only here, only BA1 was the only variant which shows a difference between this monovalent and bivalent vaccine group. And there are 2 colors here. The blue represents the non-infected one, and the red represents those subjects which were infected in between. And we also analyzed the fold change between the pre-boost and post-boost, and also compared between the vaccine type. As we can see here, the monovalent and bivalent group had similar fold increases in MBCs for Wuhan, Delta, and BA5, but bivalent group had significantly higher fold change in the BA1 group. And to detect MBCs, memory B cells binding to multiple variants, we use a Boolean gating strategy. And this Boolean gating, for the Boolean gating, the gates, we created gates for each tetramers, and then logical and combination were used to quantify cells that binds to 2 variants, 3 variants, and 4 variants. And in this graph, we can see that the number of MBCs binding to multiple variants, they were increased in both groups, but the increase was higher. The increase was higher in case of bivalent group. And this pie chart is showing the fraction of memory B cells which are binding to 2 variants, 3 variants, and 4 variants. As you can see from this, they were significantly higher in the bivalent group as compared to the monovalent groups. So now what we did was we segregated the memory B cell response based on their binding to a combination of RBDs. We made a combinations and compared between the these vaccine types. What we saw was there was a significant increase in cross-reactive memory B cell towards this Wuhan plus BA1 and Delta plus BA1 plus BA5 in the post-booster group, and it was significantly different between bivalent and monovalent vaccine. In pre-boost, there was no statistical significance. So all this analysis, it suggests that the bivalent booster result in greater cross-reactive memory B cell response in vaccinees. Now, effect of infection. Again, we wanted to see if the infection had resulted in skewed our data. So we did statistical analysis, and we found that there was no differences between the MBC values because of the infection. And the T-cell assay, The T-cell assay was done by Tony Cunningham and his group. They did a spike-specific T-cell stimulation test using fluorosport. And this was the result. What they found was, I don't know where the stars went in this. Okay, I'll just explain it. There's supposed to be some stars there. Okay, the monovalent and bivalent vaccine both significantly boosted the spike-specific interferon gamma production to the Wuhan, but only the bivalent one also boosted the IL-2 and the dual stimulated producing cells. And both vaccines induced a significant and comparable boost in interferon gamma producing T cells that were cross-reactive to BA5, but only the bivalent vaccine also induced a significant boost in response to BA1. Here, only the bivalent one was significant here, but I don't know where the star went. And then finally, we also did a correlation test between the B cell response, T cell response, and the neutralizing antibody titer. There was a significant and moderate correlation between the neutralizing titer for Wuhan, Delta, BA1, and BA5 with the cross-reactive memory B cells. And we also did a multivariate analysis, thanks to Chaturaka for helping us with that. To adjust for the effect of confounders, we tested if age, sex, prior infection, and time to blood draw since the booster dose was statistically significant between the monovalent and bivalent group. We found that sex, age, and time to post-boost blood draw were statistically significant between the groups, while having a prior infection was not. And the vaccine type independently determined the neutralizing antibody titer at post-boost against the Delta, BA1, and JN1 variants, and the memory B cell response, memory B cell count at post-boost is specific for Wuhan BA1 variants, as well as the number of MBCs capable of binding to multiple variants after adjusting for pre-boost value, age, sex, and time to blood draw. And we come to the conclusion that this study suggests that bivalent vaccine induces superior neutralizing antibody response, B cell response, and T cell response compared to the monovalent boost. And we think that the reason for increased neutralizing antibody breadth observed in the serum from individuals receiving the bivalent vaccine is due to the enrichment of broadly binding memory B cells. And therefore, the increase in broadly reactive B cells confer an advantage against the novel variants, because we had the JN1 variants which were not present at the time of infection, but the bivalent vaccine was able to produce protection against those variants also. So acknowledgements, we have a very big team and a very big acknowledgements. At first, I would like to acknowledge my supervisors, Professor Rowena Bull, Professor Nicodemus Tedla, Professor Andrew Lloyd, and thanks to Chaturaka for helping us with the statistical analysis, and Hui for all the samples and helping me with the thawing the samples and everything. Professor Tony Cunningham and his group for getting this project in place, and also with the T-cell analysis, and Associate Professor Stuart Torbill and his group for the help with the neutralizing antibody data, Professor Tony and San for help with the project, and Professor Jamie and other groups from the University of Sydney, and last but not least, Professor Stuart Tangi from the Garvan Institute. And also, this is a big group that was involved in some degree in all of this project. So thanks to them also. And thank you very much. Thank you for listening. And I'll be happy to take any question if you have.
Thanks a lot. That was fantastic talk. Any questions?
That was a great talk. It's cool to see. I remember you running all your cells and studying everything in the lab, and it's cool to see that it's come to completion. So I noticed that in your monovalent group, it looked like a split between two different vaccines. I think there'd been a couple of papers recently about how the B and T cell responses can be affected by, Moderna versus a Pfizer vaccine. And I was wondering if you took that into account in your analysis.
Actually, we had like most of them like in the monovalent group, most of them were like Pfizer group.
Oh, cool.
Only one or two were Moderna.
Okay.
Like almost all were Pfizer. So in the bivalent group, all were Moderna.
Oh, cool.
So it was very difficult to do the subset analysis because we didn't have enough sample to do the subset of Moderna versus Pfizer.
Oh, that's kind of good then. So then that's like a variable that.
Yeah. Thank you.
Hey, great talk.
Thank you.
Sorry if I missed this, but so the bivalent vaccine is a combination of Wuhan and
Omicron BA1,
right?
yeah.
So you said you have more cross-reactive B cells from the bivalent. Did you see if you had more B cells that were cross-reactive between those two variants compared to other variants?
Actually, yeah, we found that the cross-reactive variants that were more skewed toward the BA1 and also against the Wuhan, the original Wuhan variant also. So if that is what you're asking.
Yeah, yep.
Right. Thank you so much. Thank you.